In today's world, there are some things or sciences that we all need to know or at least be familiar with, because they can have a huge impact on the lives of humans. One of these sciences that we all need to know to some extent is genetics. In this video, I want to explain the principles of genetics to you in simple, completely scientific language and step by step. The most famous name in the world of genetics is an entity called chromosome, in which the information of a living being is completely stored. Any change in this entity, however small, may affect the characteristics of that living being, like humans. Let's first get to know the chromosome better. Each chromosome is formed by the combination of two biological molecules, 1. DNA molecule and 2. Proteins. DNA and protein are each types of molecules that only exist in the bodies of living beings. We call such molecules biological molecules. Biological molecules are usually large molecules and use more atoms in their construction than many of the simple molecules we know. For example, a water molecule is made up of 3 atoms, but a DNA molecule or a protein molecule is made up of more than thousands of atoms. So now we need to look more closely at the chromosome. Each chromosome contains a DNA molecule, which you usually recognize as a spiral ladder, with a lot of protein surrounding this DNA molecule, and we call the collection of this DNA and proteins a chromosome. In the structure of a chromosome, the most important thing for us genetically is the DNA molecule inside it. Do you know what it's like? Exactly like a person. Consider a person alone without clothes and nothing as a DNA molecule, and consider the clothes, watch, and everything else that comes with them as those proteins. Indeed, clothes and these accessories are basically always with a person, but what's important is the person themselves. So now let's go and get to know the DNA molecule more precisely. The molecule that encodes all the information and characteristics of every living being. DNA is a polymer, meaning it is a molecule that is made up of many smaller units linked together. In fact, there are small molecules called nucleotides, many of which are joined together to form a DNA molecule. When you link a lot of nucleotides together, you actually create a string called a polynucleotide strand. The DNA molecule is usually made up of 2 polynucleotide strands that face each other and are connected by bonds called hydrogen bonds. Then, these 2 strands that face each other twist around an imaginary longitudinal axis, creating the famous DNA structure that its discoverers called the double-stranded helix structure. The information of living beings is stored in the same small molecules that make up DNA, the nucleotides. There are 4 different types of nucleotides, and by arranging these 4 types of nucleotides in succession, different information and codes are created that can be used to determine the characteristics of a living being. Before continuing, we need to know one very important thing, which is that all information and data in the field of computer science is encoded using only 2 digits 0 and 1. But the world of genetics has 4 different letters to encode its information, and this means a very, very high volume for storing infinitely different forms of information. Well, now it's time to get more familiar with the building blocks of DNA, which are the nucleotides and have 4 different states. It's very interesting that each nucleotide, as a small unit that wants to build a larger molecule, DNA, is very complex and strange. Each nucleotide itself is made up of 3 different parts. 1. A 5-carbon sugar. 2. A nitrogenous organic base. And 3. Phosphate group or groups. The 5-carbon sugar used to make a nucleotide can be a type of sugar called ribose or another type called deoxyribose, which has one less oxygen atom than ribose in terms of its molecular structure and is one oxygen atom lighter. The nitrogenous organic base that can be used to make a nucleotide comes in many forms, but the most famous organic bases used to make nucleotides are the bases adenine, thymine, cytosine, guanine, and uracil. Based on their molecular structure, adenine and guanine are called double-ring bases or purines, and thymine, cytosine, and uracil are called single-ring bases or pyrimidines. But finally, the last part of a nucleotide is the phosphate group. 1, 2, or 3 phosphate groups may be used to make a nucleotide. Now we need to connect these 3 parts together to make a new nucleotide. Then we have to connect millions of nucleotides to make a DNA molecule. To make a nucleotide, a sugar, a base, and 1 or 2 or 3 phosphate groups must be attached to each other, but there is one condition. If the selected sugar is ribose, the thymine base will not attach to it, and if the selected sugar is deoxyribose, the uracil base will not attach to it. So, with this, many types of nucleotides can be made. Much more than 4 types. But we need to know one very important thing here. Yes, the diversity of nucleotides is much more than 4 types, but nucleotides are not only used as the building blocks of the DNA molecule. Rather, they have to do many other things inside the cell. In fact, among the various types of nucleotides, only 4 types are used to make DNA, but which ones? Let's go back a few steps. To make a DNA molecule, millions of nucleotides must be linked together to form one of the 2 strands that make up DNA. But there are several important conditions for the linking of nucleotides. 1. The sugar of all the nucleotides that are linked together must be the same. Either the sugar is all deoxyribose or the sugar is all ribose. If the sugar of the nucleotides that are linked together is all deoxyribose, the final molecule that is made will be the DNA molecule. But if the sugar of the nucleotides that are linked together is all ribose, another molecule similar to DNA is made, which we call RNA. We will talk more about this molecule in the future. 2. When nucleotides want to be linked together, they must have 1 phosphate, no more. Given these 2 conditions, we see that we have no more than 4 nucleotides left to make DNA, which, based on their bases, we call them a G, C, T for short. Now it's time to make DNA. First, we need to string the nucleotides together to make a polynucleotide strand. The nucleotides are joined together by a strong covalent bond called a phosphodiester bond, forming a chain, which you can see in different images. These pictures are all the same. But if you remember, DNA was made from 2 of these chains. So we need to make another one of these chains, but there are a few conditions for this second strand. First, it has to be in the opposite direction of the first strand we made. But the second condition is a bit more specific and complicated. To make the first strand, you can string the nucleotides together in any order you like. But when you want to make the second strand, you can't just string the nucleotides together. It must be made in such a way that the adenine base of the first strand must be in front of the thymine base of the second strand, and vice versa. That is, the adenine base of the second strand must be in front of the thymines of the first strand, and similarly, the second strand must be arranged in such a way that the cytosine and guanine bases are in front of each other. This is called a complementary relationship between the bases. That is, A is always in front of T and C is always in front of G. Now, these complementary bases form hydrogen bonds with each other, and in this way, they practically cause the two strands of DNA to stay together. The complementary relationship between the bases and the reverse orientations of the two strands of DNA are beautifully illustrated in this image. Finally, a complete DNA molecule is made. Look at the DNA molecule from this view. So far, there has only been one DNA molecule. But in the nucleus of each cell of a living being like a human, there may be several of these DNA molecules. For example, in the nucleus of each human cell, there are 46 DNA molecules, each resembling a long spiral ladder, and these 46 have encoded all our information in their nucleotides. Of these 46 DNA molecules, we inherited 23 from our father and 23 from our mother. We will discuss this inheritance issue in more detail in the future. Oh, let me tell you a historical fact about the discovery of DNA. In 1869, a Swiss scientist named Miescher extracted a substance from the nucleus of fish sperm cells and human white blood cells that had weak acidic properties and named it nucleic acid, meaning an acid extracted from the cell nucleus. Finally, when the process of discovering this molecule was completed, due to the type of sugar used in its nucleotide, which was deoxyribose, its full name was deoxyribose nucleic acid, or DNA. But let's get to the proteins that are next to DNA. Of course, there are many proteins that are with DNA, but a group of them are very important, called histones. Now, what do these histones do? First, I have to say something that will surprise you very much. Each DNA molecule in the nucleus of every human cell is about 5 centimeters long. If we multiply this by 46, the number of DNA molecules in the nucleus of human cells, it adds up to more than 2 meters. That means that in the nucleus of every human cell, whose diameter may not even reach 20 micrometers, there are more than 2 meters of DNA molecules. So how does it fit in? By twisting around histones. The DNA molecule wraps around 8 histone molecules about 2 times in several places, and thus becomes a little more compact and shorter. Imagine how much shorter and more compact it becomes if DNA is twisted in several places like this. And finally, it fits in the nucleus of the cell like this. Another interesting thing is that the cell can increase or decrease the amount of this contraction or compression, and this depends on the needs and conditions of the cell. But anyway, if these histones were not there, the DNA molecule would be very tangled in the cell nucleus and its information could not be used at all. Imagine how chaotic and chaotic. Finally, I want to tell you about how our information is stored in the DNA molecule. In fact, information is determined and encoded according to the order and sequence of organic bases of nucleotides. For example, this order of organic bases has a specific meaning in the cell and body of a living being, and if this order changes, its meaning also changes. It is clear that the more similar the sequence of organic bases in 2 living beings is, the more similar the 2 beings will be. It may be interesting to know that the sequence of organic bases in all human DNA molecules, compared to the total organic bases in chimpanzee DNA molecules, shows a similarity of more than 98% between these 2 beings, which is really strange and interesting. But I must also tell you that scientists believe that there are parts of the DNA molecule and sequences of organic bases in DNA that do not seem to encode specific information, or at least we have not yet discovered the information in those parts. However, here is an important definition that we need to complete our discussion in this video with. Parts of the DNA molecule whose organic base sequences have a specific meaning for the cell or living organism are called genes, and other parts that do not seem to encode specific information are called intergenic regions. In the next videos, we will talk more about genes and their functions, and we will also learn the principles of heredity together. See you.